Let's start. About 50 or so have registered. I hope they'll be joining soon. And we'll start now. So, welcome all to yet another webinar of Kerala Biotechnology Commission. As you all know, Kerala Biotechnology Commission, functioning under the Kerala State Council for Science, Technology and Environment, caters to the development of biotechnology activities in the state of Kerala. And at times we organize the different webinars by renowned biotechnologists throughout the country and the world. And this is one among them. And we have, before going to introduce the speaker, let me welcome our distinguished speaker, Dr. Arumugam Rajavelu from IIT Madras. And I also welcome my colleague, senior colleague, Dr. C. Arunan, who has joined now, and all the participants to this webinar. I hope you all have a wonderful time. Let me now introduce to you Dr. Arumugam Rajavelu. He is currently the assistant professor at Department of Biotechnology, IIT Madras. Recently joined at IIT, and started his research career as junior research fellow at Indian Institute of Science, Department of Biotechnology, and then subsequently moved to Germany with a PhD fellowship funded by German Research Foundation in the year 2008. And then he has got the postdoctoral fellowship from Stuttgart University, funded by German Research Foundation, Germany. And he's an awardee of DST Inspire Faculty Award instituted by the Department of Science and Technology, Government of India. And he has joined Rajiv Gandhi Center for Biotechnology as the Inspire Faculty Fellow in the year 2013. And he was there till recently, at Rajiv Gandhi Center for Biotechnology. And we are proud to say that he's the awardee of Kerala State Young Scientist Award, and also awardee of Young Investigators Program in Biotechnology instituted by Kerala Biotechnology Commission. And he is also awarded with the Young Scientist Medal from Indian Society for Parasitology, young dynamic researcher, specialized in the area of malarial epigenetics, particularly onto understanding the chromatin plasticity of human pathogen, the Plasmodium falciparum, and his research interest being the molecular epigenetics and plasmodium biology, cardiovascular diseases, and diabetes biology. And he has published about more than 50 international peer-reviewed publications, and as I've already told, several awards. With this, let me invite Dr. Arumugam for the invited talk. Over to you.
Thank you. Let me share a slide first.
Yes, sure.
Is it visible now?
Yes, it's getting loaded. Yeah, loaded. You can make it full screen. You're fine.
Okay.
Fine.
So, first of all, let me thank the organizer, Kerala Biotechnology Commission, and Dr. Sarika for a detailed introduction about me. And thank you for the invitation to present our work briefly in this webinar series. Today, I'm going to present one of our recent work and also briefly explain the epigenetic signatures and various pathogens, how these signatures are essential for the pathogen to survive and to initiate an infection, very briefly. Yeah, so I thought I'll just start with this picture. This is a Department of Biotechnology where I joined very recently, as Sarika introduced, in July 2021, just 4 months before I joined here. And this is the IIT campus, one of the most beautiful IIT campus in the country. You will see, you will live within the, like you feel like you're living within the forest. The deer is always around, deer and monkey. And so very interesting campus. And I thought I'd just show you the list of few microorganisms selectively. So why these microorganisms? As you all know, a few microbes are free living. It doesn't depend on the host. But when a host is there, it just start infect the host. There are few microorganisms which are very obligate, which strictly multiplies, lives within the cell or eukaryotic cell. So here I brought up both the cases. For example, Mycobacterium tuberculosis, one of the important pathogen which causes the tuberculosis, which is a free living ulcer because it can be cultivatable, but also it will multiply within the cells. Whereas the protozoan parasite, it's an obligate parasite. It will not multiply outside the cells. It will multiply only the inside the cells. So, what we know past few years about these intracellular pathogens, you might have come across these intracellular pathogens primarily utilizes the host factors, including viruses. Viruses will not replicate without host, as you all know. It will utilize the host factor and multiplies. So host factor means primarily we always think about it most likely the proteins or amino acids, degraded proteins, products, that's amino acids or minerals and so on, or is ions from the host. So what I'm going to explain today, how these intracellular pathogens modulates the key factor, that's how it modulates the genome level or expression of the host genes for its survival, and primarily how these pathogen modulates the epigenetic machinery for its survival. And since I'm going to talk about detailed about the epigenetic machinery for newcomers, I thought I'll just briefly introduce what is epigenetics. I will just try to show with an example from the human perspective. Human body, as you all know, we have nearly 100 trillion cells. These cells are, are formed into almost nearly 200 different cell or tissue types in our body. So, as you all know, this each and every cell has the same genetic information. You take any cell in our body, it has a 23 pairs of chromosome, same genetic sequence arrangement. And depends on the cellular context, tissue context, you have the only selective subset of expression in each and every cell. So, if you look at, we have the same genetic information in each and every tissues, but you have the different functional, for example, different tissues, brain tissue, skin tissue, bone tissue, muscle tissue. You take out the genome sequence of muscle and bone and then liver, you have the same. But how these become very, completely drastic phenotypic cellular structures. So this is the questions people they started to ask this, and then finally identified there is a factors called an epigenetic factor, which play a key role in deciding the outcome of cellular fate. For example, the genetic information must be expressed at right time, at right place. There is any misregulation in the gene in this expression of these genes in these different cells, you have an apparent cellular phenotype, and that is how now it's connected very well with many of the cardiovascular diseases or even the many cancers. When you take any cancers, you'll have an aberrant epigenome, which clearly says that the proper epigenetic network or modifications are essential for the successful development. So coming back to the bottom, when cells develop from the stem cells to the multicellular level, so there is this kind of orchestrated, programmed gene expression pattern, which dictates to become a more functionalized tissue or cells. So that more orchestrated programmed gene expression is tightly regulated by the factors called in epigenetics, epigenetic players. What is epigenetics? So it's a branch of biology which deals on how interaction between the genes which creates a stable trajectories for development. This is a textbook definition. To make it more simpler to the common audience, I would like to say that it's a casual interaction between a genes and environment, which changes gene expression without altering the DNA or nucleic acid sequences. Most importantly, these changes are heritable. This is called an epigenetics. When more simplest way one can define epigenetic, there is a changes in the gene expression in a tissue or a cell in the response to the environment, but there is no changes in the DNA sequence. So when you have a changes in the gene expression, you always think there must be a changes in the DNA sequence. No, and in this case, on top of the DNA sequences, there is some modification actually, which these modification in response to the environment decides that the particular gene to be expressed or not. So that's the basics about an epigenetics, and he's the man who coined the term an epigenetics, Waddington. So imagine he coined the term in much earlier, almost 1950 itself. So that time itself, the concept of epigenetics emerged, almost 70 years before itself. But the understanding about molecular epigenetic came much later, almost after Human Genome Project. So he explained the epigenetics using this simple picture. He explains you roll a ball on a valley. Okay, this is a valley, and there is a 4 valley here. And when the ball rolls from the top to down, and then once it commit to travel here, and then ball goes here, and then travels. Then the ball cannot jump from between the one valley to another valley. So he connect this theory and this model to the cellular development. When a cell develops from the stem cells, from the zygote, once it commit to the particular functional tissue, when the cell develops and commit to the particular the blood cells or liver cells, and then after that, this will not go back to the another cell types. So that's what he tried to explain. That means there is a strong and an epigenetic program operated when the cell develops and towards a particular functional tissues. So what is one should keep in mind, the precise and epigenetic network or players are very important in controlling the whole cellular development for a successful growth of organisms. So these are the an epigenetic plays which I want to just explain. So this is an human genome. You can see this is how the human gene is organized in the nucleus. You have a DNA structure which is wrapped on the histone proteins, and then these histone proteins form histone proteins, the DNA form like nucleosome structures, and then condense to become a chromatin, and then chromatin fiber, and then chromatid, and then chromosome structures. So if you remember the basics, we have each and every cell, we have nearly 2 meter DNA. If you take out all 23 pairs of chromosome DNA structures, if you elongate on the table, the DNA length will be nearly 2 meter. And our cellular nuclear size is very small. It's nearly 5 to 8 micron maximum, or even 10. That's it, not more than that nucleus size. So the 2 meter of DNA structures need to be packed precisely into such a small nuclear structures, right? So such packing mechanisms is essential to cope up or to keep the large genome size in our body. So how this is being achieved? The complex network of chromatin remodeling, which is correctly organized by the epigenetic players, which is essential for the organization of this DNA into the nucleus. So this is how DNA structure is packed on a histone protein, and then the 3 important epigenetic players which involved in controlling the whole process which I was explaining. One is on a DNA methylation, and second an histone methylation. Histone is a protein, a basic protein, which is present here. On top of the basic protein only, you have the DNA structures wrapped. And now very recently it is reported a few set of long non-coding RNA also involved in this process. So how this is involved? It actually involved in recruiting a protein which introduces a modification on DNA or histone protein, on histone proteins. So, but primarily these are the 2 key in epigenetic players. One is on DNA methylation on a cytosine base. You have the 4 bases: adenine, guanine, cytosine, thymine. On a cytosine base, there is a modification, and these modifications are involved in an epigenetic process. And histone methylation on either lysine or arginine amino acids on histone protein, and mediates specialized gene expressions in each and every cells. So this is an epigenetic modifications. Epi means above, genetic. On above to the genetic material, there is a changes or a modifications. It's called an epigenetic modification. And now more recently, there is another modification was discovered. Even though it was kind of reported very early, but its connection to epigenetic or epigenetic process linked very recently. Those are called epitranscriptomic modification. You have a modification on the RNA, that's a methylation on RNA, which is involved in stability and regulation of the protein synthesis in the cells. So this is called an epitranscriptome modification. So these are the major players control the whole process of different cellular gene expression in the various tissues. Okay, so whether these modifications are conserved. So before I talk about this, let me make it very clear. The concept of epigenetics or epigenetic modifications are highly restricted only to the eukaryotic organisms. I mean to say from the lower eukaryotes to the higher or multicellular organisms. So the epigenetic process kind of not well studied or not well conserved in the prokaryotes in case of bacteria. Even though the DNA modifications are reported, the same DNA like cytosine modifications or methylations or adenine modifications is present on the genome of bacteria, but that's not linked with the epigenetic process. So that's why I just took an example only for eukaryotic organisms. So the DNA methylation, histone methylation, long non-encoding RNA, which is conserved among the lower organisms, from unicellular organisms to multicellular organisms. So most of the modifications are conserved. DNA modifications are a little bit conserved. There are few exceptions which I'll show you very shortly. And then a long non-coding RNA also conserved across the species, which says clearly, irrespective of the multicellularity or the unicellular nature. So the basics of epigenetic modification is essential for a successful gene expression as well as the growth of organisms. Yeah, so this is a few examples which I want to highlight here. DNA modification or methylation is an important epigenetic modifications, but a few lower eukaryotic organisms which does not have these modifications, which is extremely dependent only on a histone methylation modifications. For example, Saccharomyces cerevisiae and then Schistosaccharomyces pombe is a budding yeast and fission yeast. These are two model organisms which is used extensively for gene expression studies. Does not have a DNA methylations. Similarly, the Drosophila, fruit fly, and then C. elegans, and then Dicty. So these are a few model organisms essentially used for many gene expression analysis, but this does not have a DNA methylations. But it retains the long non-coding RNA activity as well as the histone modification. So coming back to the prokaryotes, even though I say the prokaryotes does not have an epigenetic signature, classical epigenetic signature, but the genome sequencing of those few pathogens of prokaryotes organisms identified, it indeed encode a histone lysine methyl transferases. Then obviously question comes, see, the bacteria does not have a chromatin. Again, it has only the DNA structures, does not have the histone protein, does not have a structure like other eukaryotes, higher eukaryotes. So then why this genome should encode a histone lysine methyl transferase when it does not have histone? So then this, scientists started to investigate and identified. So these are the few microorganisms already they reported, Mycobacterium, Listeria, and Chlamydia, and so on. And they found these microorganisms encodes these histone lysine methyl transferases, which actually induces the histone modifications. And then these proteins become a secretory nature here. And these enzymes are non-secretory nature in eukaryotes, But when the bacteria encodes, this becomes secretory. That means it just is produced in the bacteria and comes out of the bacteria and then goes inside the nucleus and modulates the chromatin structure of the host cells. So you see how cleverly bacteria evolved. It brings its own methyl transferases to modify the host chromatin and then changes the host gene expression pattern for its survival. It is even reported it modulates the expression such a way that it can escape from the host cell immunity and so on. And an example here, I'm just trying to give an attention for you, Legionella. So this is one of the kind of very top paper appeared, and in this case, what they found out, these bacteria secrete a protein that's histone lysin methyltransferase. What does it do? It does not methylate on a regular positions. There are regular positions or canonical positions on a histone protein, but this enzyme doesn't methylate on a regular positions. Rather, it methylates on a unique positions and in histone 3 proteins and a specific lysin, which usually does not get methylated in humans or any other eukaryotes. But this bacteria enzyme does it. So this, in a way, this stands unique to modulate the host systems. And similarly, Mycobacterium tuberculosis, it has it encodes many enzymes, HAT, PKMT, and PRMT, and so on. And these enzymes basically modulates the host genome and for its better survival. And particularly, it is reported Mycobacterium tuberculosis enzymes modulates the host genome to survive inside during the hypoxic condition and so on in the macrophages of the lungs. Yes, so viruses, you take in any viruses or many virals, it is reported either its viral genome gets modified by the host vector because it does not get genome is too small, does not encode sorry, does not encode histone lysin methyltransferases or DNA methyltransferases, but its genome gets modified by the host vectors. Or there are few reports, many viruses, in a few viruses, it is shown that the virus, through or directly through network, it alters the host epigenome. But again, many things need to be discovered here. But since there is an ongoing pandemic, we have written on nice review very recently from our group. We put our own perspective. What we have put here, as we all knew from many recent outbreak, many informations, the SARS-CoV-2 has the severity among the kind of chronic diseased individuals. If you look at overall mortality rate among the chronically diseased individuals, the severity is very high with the SARS-CoV infected individuals than the normal individuals. Then obviously question comes, why these chronic individual patients are more prone or they develop the most severe cases. So when we collected many literature and we found out there is the common pattern among the chronic diseased individual. You take any of the chronic diseased patients, hypertension, heart disease, systemic erythematitis, any patient if you take it, it is evident that there is an aberrant signatures, an epigenetic signature in these chronic diseased individuals, and it is not clear yet whether these really contribute. But we proposed a model, this aberrant epigenetic signature of the host in favor for the increased replications or severity of the SARS-CoV infections. But anyway, these need to be investigated whether this truly contributes. But anyway, this is a new dimension which we put it got recently accepted. So so these are the few general epigenetic perspective from various bacteria and viruses and so on. But now I'll just try to take it to our work. So what we are doing right now. So we work on another human pathogen, that's intracellular pathogen, and Plasmodium falciparum, that's which causes the malaria. It's a protozoan parasite. So just to refresh a little bit about malaria disease, even though you read many textbooks, it's another major disease in the tropical countries. If you look at here, most of the tropical countries which is high with the rich in a mosquito, you have the eye transmission of the malaria disease. Nearly 229 million cases are recorded every year. This is as per the WHO, and of which almost 500,000 deaths are recorded every year. And preferably the children are the most vulnerable to this malaria disease. What is the more alarming? Even though there is one recently a vaccine approved by WHO, but there is no good vaccines are available against the disease. For this, there is multiple reasons. The parasite is very complicated and is obligate intracellular. It lives only within the cell, and which keep on change the surface protein on the surface. So there are multi factors contributed for this failure in the vaccine development. Only option so far successful in controlling the malaria through the anti-malarials. The early times it was treated with the chloroquine. Later it was stopped due to the resistance. From 1995 onwards, in the clinical scenario, it was heavily started treating with the malaria patients were treated with the artemisinin and its combination therapy. It was very successful, but the problem started in almost 2014. By the time when the malaria cases started declining from 2010 onwards, the parasites started developing resistance in the Southeast Asians against the drug. So this is the only drug available, and then to this drug also the parasites started developing resistance, which again clearly says that still we need to have a better understanding about this parasite to develop the unique drug molecule to control the parasite. So this is the life cycle of the parasite. The life cycle, the parasite multiplies in two hosts. One is mosquito and human host. Mosquito is a definitive host where the sexual multiplication takes place. So mosquito releases the sporozoite when it bites during the blood meal, and then sporozoite travel to the liver, and from the liver it multiplies and modifies its phenotypes and releases the merozoites. Then this is the one form, small dot-like structures, and this merozoite infect the RBCs. So this is the place you will have the clinical symptoms. In RBC stage, that's a red blood cell stage. It goes three different forms: ring, trophozoite, and schizon. So the same parasite, you see the phenotypes, three drastic phenotypic structures when it develops into the RBCs. So the whole cycle completes in roughly 36 to 48 hours. So when one RBC infected with one parasite, and then within 48 hours, it will produce nearly 30 parasites. So because of the cycle nature only, you'll have the fever and chillness symptoms in the malaria cases. So when this keep on develops, and when it is not treated with an anti-malarial drug, what happens? The parasite spreads throughout the body and different organs in the bloodstream, and then when it goes to this brain also, this because of this surface protein expressed by the parasite, it just starts sticking onto the blood vessels and causes the cerebral malaria. So this is one of the severe form of malaria which will lead to the high mortality among the infected individuals. So in some cases, what happens when the parasite develops in the RBCs, few undergo to the gametocytes, and then again when the mosquito bites, the blood it takes it. So this is how the cycle continues. So what I want to bring an attention for you. So from the release from the mosquito and then from the gametocyte, if you look at the parasite structures, it's not a single. If you take a bacteria, you have the common phenotypes. When you take even like yeast or fungus, you have the kind of single phenotype structures. But in this case, you have multiple phenotypes. If you take it, almost nearly 7 or 8 different phenotype structures by single parasite are displayed in the blood. That's why one of the very challenging to control this disease by developing a specific vaccine. So obviously, question comes here. Yeah, it has a very small genome. So how these different phenotypes are produced, and who control these different phenotypic, differential gene expression in these various phenotypes? So again, I want to bring just refresh a comparison between the. Are there any differences in the epigenome or epigenetic structures between the parasite and host cell in which it lives? So this is also, as I said, lower eukaryotes, unicellular organisms. It has a conserved histone methylations. there are few differences which I am going to present today. And then DNA methylation in the parasite, it was a questionable. We reported this parasite does not have a DNA methylation like other model organisms. And then most importantly, it does not have this, this is not a strong epigenetic machinery, but it's still it participates a little bit in epigenetic network. Histone H1 proteins and RNA pathway, which is partly involved in a small epigenetic process in higher eukaryotes, which is absent in the parasite, which tells that the parasite has a very unique an epigenetic signature comparing to the other eukaryotic organism, or the organism the organism in which it lives. For example, in human, compared to the human cells, the parasite, it has a contrast or a distinct epigenetic signature, it seems. So in one example where we are shown this enzyme DNA methyl transferase like enzymes are present in the parasite, but actually it does not methylate DNA, but rather it methylates the RNA structures, which is again pretty new or quite unique to the parasite. And then similarly, what happened, the parasite, I'm talking about epitranscriptome of the parasite, is slightly different than the human cells, human systems or human cells. So in epitranscriptome modifications, you have the kind of mRNA which gets modified. For these should be recognized by these, the specialized protein called YTH domain like protein, which bind, and then it decides the fate of the RNA. So that means the whole process complete, you should have the three factors: enzyme, that is a writer that should have the modification here, and then a reader that should bind here, and then eraser. Actually, that's a demethylases that should remove the modification to bring the whole dynamics of the epitranscriptome of any organisms. So this is conserved in all the species: reader, writer, eraser. That means an enzyme which adds protein which bind to the modification, or enzyme which removes the modification. But in a parasite, only two is present: enzyme which adds, and then reader which binds. But eraser, the genome does not encode. Again, it stands unique. So which does not depend on the eraser, but fully depends only on the enzyme which adds as well as the protein which bind to the modification, and through which the parasite genome or mRNA gets regulated. So in this way also this parasite stands unique. Okay, so just to refresh here how the parasite completes its life cycle in as RBCs. Again, to reiterate, it has a many phenotypes. If you look at, you have complete different contrast structures. And then it has a nearly almost 22 MB genome size, which is very small. And these are distributed as a 14 chromosomes in the parasite. And just imagine when the parasite developed from this stage, this 14 chromosome has to be carefully organized. And then when it goes to this Kaizen stage, that all the 14 chromosome has to be asynchronously divided and distributed into this another all small parasite. So that should be a complex programming must be happening in this case in order to complete the life cycle of the parasite. So this is the place we were interested. So who control this complex process of gene expressions? If we identified these complex process of gene expression, then it's easy for us to design a very good drug molecule against these parasites. So we have a hypothesis that as the parasite does not have many transcription factors, and then the tight gene regulation of this in the parasite between these stages must be largely depends on the only chromatin modification or nature of the chromatin structures of the parasite. And then we were thinking the parasite may have a unique or a kind of a very novel modification on the genome, and these modification may involve in regulating the gene expression at different stages. So this was an idea which we started, and then one of my PhD students started work on this project, and then we started to identify modifications on this parasite histone protein. So I want to just again to refresh a little bit here. So this is how the nucleosome structure is present in any eukaryote. You take it from any lower eukaryote to the higher eukaryote. You have a DNA structure wrapped, and then you have 4 histone protein, which is in the duplicate. Totally 8 proteins will present, octamer. And then the proteins, each histone proteins will have a tail. Okay. So the tail protein also gets modified, as well as the histone core also gets modified. So each has a different functions. So in our case, we started to look at only the modification on the core regions. We were not interested to identify these modifications in the parasite, as these modifications are well conserved across the eukaryotic species. So we thought we start to look at the unconventional modifications on the only core regions. Okay, so we started to culture the parasite, that we could able to do that, and then my students, they performed to extract histone proteins, and they did histone they extracted, and then we identified a modification on 3 different positions. One is histone 3 and arginine 42 and lysine 64, and another histone 4 lysine 44. These are the different modification, which is very rarely reported in many species, which is seems to be very unique to the parasite as well. So we started to characterize, but I'm going to show data on only the one modification very briefly, and which is a very novel to the parasite as well. So this is the mass spectrometry data. This is through which we identified. This is a peptide which I identified, and then that has some modifications. That is a trimethylations on this lysine, which is equivalent to the each methyl group, the 14 Dalton size, and this difference is equivalent to the 3 methyl group. And then later we confirmed this modification by immunoblot. And yes, when we isolate the histones or nucleosome from the parasite, and then you could able to detect the modification. So later we were thinking, okay, so this, since epigenetic modifications are strongly influenced by the environment, we were thinking whether the culture dependent adaptation, whether this modification just acquired due to the culture conditions. Then we did a Western blot on a clinical isolate, and then we got a similar signal, so which confirms this modification must be having the physiological relevance. Then we mapped where this modification is present on nucleosome, and we found that this is very closer to the DNA where the structure is wrapped. Okay, so which implies this modification may have a role in directly influencing the DNA wrapping on the histone proteins, so which need to be investigated. So my student again started to identify an enzyme which is a very new, because even though there is a one report available for this modification earlier, but there is a no enzyme has been reported so far against this specific and epigenetic modification. So we were able to clone a complete set of enzyme, and then we set up a biochemical assay to identify an enzyme responsible for this modification. There is a large set of data assay I'm not going to explain, but I'll just briefly show you. This is autoradiography assay, which shows all enzymes are active, and then immunoblot assay confirms the 2 enzymes are specifically methylating on these positions. And then another sequence of biochemical experiments by my student confirms these proteins actually selectively methylates the intact structures. What are the intact structures? When you give the histone protein alone, does not methylate. Okay. When you give the full nucleosome, I mean histone protein wrapped with the nucleosome, so this is the favorite substrate for these enzymes, which says again, this enzyme has some interaction to the nucleosomes. So we model these through structures, and we found there's a selective interaction only to the DNA, not to the histone protein. That means it interacts with the DNA structure, it may get a conformation changes, and then it methylates. So this is a mechanism of action of this enzyme. And then we also confirmed by some other assay when we are trying to remove these DNA structures by treating with the enzyme, and we methylate using the enzyme, but we could not able to methylate, which says clearly the intact nucleosome is essential for the methylation activity by this enzyme. And the next question we asked is, was this modification is stable when it develops? ring tropocyte schizon, or it is kind of any dynamic. When we tested, compared to the other two modifications, which is a kind of well-known conventional modifications, this is the modification which we identified, and we found when a parasite develops from one stage, that's a ring tropozoite and schizon, and then you have the decreased methylation mark in the schizon, which implies that it has some meaning in the regulation of the gene expression in this particular stage. And then we confirmed this dynamic distribution using immunolocalization, which again clearly shows that the mark reduced when the parasite grows in the third stage. I'm talking about these three stages. These three stages are clinically relevant. And then we did a global chip sequencing assay, analysis for this modification to map where this modification is present on the genome level. And then we have found that the distribution is exactly matching like with the Western blot. You can see the chromosomal mapping is more with the ring and tropozoite, and then when it goes to schizon, the chip reads are very less in schizon. So it confirms that this modification is highly present in the ring and tropozoite. Sorry. Then we looked at in the ring and tropozoite, and which are the gene actually it highly enriches. When we looked very closely, we found there are very selectively these modifications are present on a few genes which encode for exported proteins. So earlier it is now when the parasite grows in RBCs, ring and tropozoite and schizon, in this case, and ring, tropozoite and schizon, and then the exported family protein, that means the proteins are expressed by the parasite, comes out of the RBC and remodel the RBC here, and then rupture the RBC and moves out, right? So people reported actually it expresses, but how this is being expressed only in this stage, it was largely unknown. So it nicely connected with our finding. These modifications strongly correlated with the export family of genes. And then when we looked at expression data sets from the public data sets reported by many other groups, we have found there is an anti-correlation between this epigenetic mark and the schizon specific gene expressions. So to summarize this part here briefly, so when the parasite develop ring and tropozoite and schizon, okay, so then it later releases the merozoite, and then the merozoite start ring. So this is how the cycle continues, right? So it is now, and in the third stage, the proteins largely exported from the parasite to the cytosol. That's called export family protein. And then these proteins are essential to remodel the RBCs, and to remodel the RBCs, then rupture the RBC to release a merozoite. This is known, but how this is being done and who controlled it, it was unknown. So since we identified a new epigenetic modification, and then that it's very nicely connected here, and when the modification comes down dynamically, just lost here in this stage, it allows the upregulation of the external protein here. So which is well correlated here. Okay, so this is what I was trying to explain. So the parasite exports protein to the RBC cytosol that helps the protein trafficking and remodel the RBCs in this third stage, and for its survival and to rupture to release the merozoite. So the for parasite to complete its cycle, so this process is essential. So here I want to reiterate or highlight here. The parasite evolved with a novel epigenetic modification, that is estrone 3 lysine 64, and these modifications are involved in controlling the stage-specific gene expressions. Okay, so I was telling you, right, it has a large genome set, right? Sorry, it has a 14 chromosome which is distributed. It is carefully. It has to be organized in 3 stages, and then that means its chromatin structure also need to be carefully organized. So now you can think about it. So this modification is present on a nucleosome structures, which is very near to the DNA wrapping regions. So which directly tells that this modification may influence the DNA wrapping nature and thereby influencing the expression or compaction or relaxing of the chromatin that need to be investigated and may help for the stage-specific gene expression. And that's what is happening in this case as well. Okay, so what is the application? So this is a kind of discovery study what we have reported right now. It is published in a very good journal. But we were thinking, this can be nicely utilized as an ideal drug target, as two enzymes evolved to methylate some novel proteins, and this developing a good molecule, inhibitor molecule against this enzyme can be effectively used to control the and parasite growth, and anyway that need to be tested. So what I want to conclude overall, in connection with the intracellular pathogens. So uh, I mean, it is not clear whether when these intracellular pathogens are parasites, or whether it has its own epigenetic machinery. Okay, so one can imagine the lower eukaryotes has the chromatin structures. Okay, and that should have the epigenetic machinery. But why bacteria should have the epigenetic machinery or epigenetic enzyme in its genome? So maybe during its evolution, maybe whether it is a co-evolved along with this interaction with these different species, that is unknown. Or it is very specially it is in response to the when respond to the host or with a non-host condition it is evolved. That is again it is not clear here. So what is clear evident as of now? The intracellular pathogens are evolved with a unique and epigenetic signature for its survival in the various eukaryotic host, and then it keeps multiply and initiate the infections. So, so with this, I want to bring end of my talk. I want to acknowledge a few funding agencies, particularly the study supported by the SERB, DST, Department of Science and Technology, and then of course a startup fund from RGCB, my former institute, Trivandrum. And then, as Sarika introduced, I got a initial funding from a good support from KCST, Kerala Biotechnology Commission, and I'm very thankful for that. And now, we are thankful to the IIT Madras for the good startup to initiate our work here, to do the follow-up study and what we are doing right now. So this is the team in RGCB, and you can see probably some of the faces are familiar for you guys. Dave, Jabina, Gayatri. So what the part of the work which I presented mostly from Jabina, and then they are successfully graduating now. So and then two JRF, Aparna and Dakshmi. So with this I end here, and very happy to take up any questions.
Thank you so much, Dr. Arumugam. It was great listening to you on epigenetic signatures and how it remodels the host cells for their survival. And now I request the participants to ask any questions. If you have, you can unmute and then ask. Any questions?
Or if you feel, or if you're feeling if you're hesitating, also you can always write to me. Okay, you have my email ID, and then I'm very happy to clarify. But still, if you have questions, please.
Yeah, you can also post in the chat box.
Yes.
I think so many students have joined. No questions? Otherwise, for the sake of the students, can you please explain your journey of research in biotechnology, so that it can be a motivation to?
Yeah. Maybe I should have. I feel now maybe I should have taken even. Maybe I thought maybe very fundamental level I should have touched it. I thought giving this new dimension on a plasmodium epigenetic would be interesting. But it's fine. So as Sarika introduced, I started my educations in my hometown, Dharmaburi. That's near to Salem, if you know about it. So that's a place I did my bachelor's in microbiology. Then Chennai for MSc microbiology, and Madras University. Then after the masters, and then I got an opportunity in an Indians of Science, the Department of Biochemistry, where I worked as a junior research fellow almost 3 years. So after that, I got an opportunity to work on an epigenetics. So I started to learn an epigenetics during my PhD time. So where we were working on the fundamentals, we didn't work on any disease model or organism and so on. We work how various epigenetic players are involved in the gene expression, various tissues and so on. Very basic level we were started to work on in my PhD work in Germany. And then in fact, I continued with my same professor in postdoc as well, even though it's a different university, but it's the same lab, because he moved to the new laboratory as a director, and then I just moved along with him. And then the start postdoc, I got an opportunity to return back. So usually there is a trend. You always come and start with what you are performing in a postdoc. Okay, so when I decided to return back to India, I thought I'll just start with my own niche. We were not. I'm not experienced in a PhD and postdoc on a malaria parasite. So, but I know some cultural methods a little bit, but not in detail. So I thought I'll just take a challenge and start propose new on the pathogens to work on an epigenetic of the parasite and pathogens. So that's how I started to propose, and we got a good funding. And then I joined in Arumugam Rajavelu for Biotechnology 2013 December. It's almost 8 years now, nearly. So I worked there, slowly built the group, and then I got a good support, including Kerala Biotechnology Commission. I was very happy. Our work was recognized by this KCST, and I got an award, Young Scientist Award from Kerala Biotechnology, KCST. It was slowly, we built, and even though we put a lot of effort here, but most of the credit, goes to the students. I want to take a challenging questions, but they traveled along with me, and they made this to such a success level. So that's why the whole credit goes to the students. So their complete dedicated work, which we could see that we really build a nice questions. I presented one dimension of a work. There are the 3 or 4 good direction of a work still is pending, which we are parallelly working on it. It's very interesting actually. So we started with a very fundamental level. Now we started understanding how this epigenetic machinery really contribute in the parasite pathogenesis biology, and whether we can utilize these epigenetic machinery of the parasite for the controlling of the malaria and so on. So that's the next level we are asking this question. So that is how I ended here. So basically, I trained in a biochemistry, microbiology and biochemistry, but in a research questions level, I ended in epigenetics.
Thank you so much.
Okay.
Thank you once again for the full vote of thanks. I invite Arjun Prasad, the scientist of KCST.
Hi, Sharika. Am I audible?
Yes, yes.
Okay. Good morning to one and all. And it was really interesting to listen to Dr. Arumugam in the field of epigenetics, actually. So I think through his example of the malarial parasite, I think the field of epigenetics, how important it is in drug discovery and related applications is very well said by Dr. Arumugam. I just have one question. I don't know whether I have missed it during your talk. As you have mentioned in your talk, the vaccine, one of the vaccine for malaria was invented recently. So, according to you, what is the main hurdle, or what are the main hurdles in developing a vaccine for malaria?
Yeah, so there are quite a bit of reasons. The main reason, it is the obligate intracellular parasite. Okay, so a vaccine, if you logically, if you think, you immunize it and you build an immune response in the body. and the pathogen comes out, you directly whether T cell or B cell, attacks. But this case, it lives within the host cell, like it goes and multiplies within the liver cell, and then it lives within the RBCs. So never exposed to the directly to the bloodstream directly. So that is one of the challenging for our immune system to recognize the parasite because of the obligate intracellular nature. But still, what happens? This parasite expresses a few proteins on the surface of the host cells. For example, when it lives, multiplies within the RBCs, it produces a few proteins which comes out of the surfaces. So there are scientists were aiming to, okay, we can develop a vaccine. We can use those proteins as a potential vaccine candidate so that we can eliminate the infected RBC alone. Again, the parasite has an kind of very unique mechanisms. So when they were developing vaccine against this candidate, so later it was found that this parasite keep on switching those proteins. That's called antigenic switching. So because of these two nature, obligate intracellular nature and then antigenic switching nature, it is very challenging to develop a vaccine against blood stage. Okay. I guess there was a combination, like there is another stage when the mosquito bites, that's a sporozoite stage. So that stage they were aiming to capture, but again it's very challenging. And then there is a third stage from Skyzone, when merozoite releases, and there is a large amount of circumcellular protein. So there are different regions, scientists are working out to identify the best candidates. But because of these two important nature, it has become very challenging. And the current model also, it is giving some extent, yes, in a smaller population. But we need to see in a larger perspective how this will help actually. Earlier also there is a hype on a vaccine against malaria, but it's flopped because of this. So what is known as of today, we are successful in controlling malaria only with the anti-malarials, not with vaccines. So still identifying anti-malarial will be the more valuable than working towards vaccine.
Okay.
Because of the complex nature of the parasite.
Okay. I think that's really challenging, and I think the inputs from the field of epigenetics and related can definitely provide good inputs in that regard, isn't it?
Yeah, so we have provided basic information. So yeah, so when keep work and keep move on, we will understand better.
Okay, thank you. So on behalf of KCST and all the participants joined for today's webinar, I would like to express my sincere gratitude to you, Dr. Arumugam, for your very interesting talk on epigenetics. Thank you very much.
It's my pleasure. And thank you. Thank you all.
Our sincere gratitude to all the participants joined for this webinar. Thank you. Over to you, Sharika.
Thank you once again, Dr. Arumugam.
Thank you. Okay. Bye to all.
Bye.